Oh. Whoa. Oh my gosh. I'm going to need to... Okay. Boink. Okay, just getting everything set up. Boop. Oh gosh. Oh. There we go. There we go. Okay. Ugh. I can't right now. Okay. Hello. So, welcome to a new series that I'm going to be calling The Art of. And The Art of is basically showing pretty much the art that is behind something. And what this art is going to be, it's going to be the art of developmental biology. And the only reason I'm doing this is because I need to study for my developmental biology exam, which the developmental biology exam, it's on Friday, but I'm going to be studying for it. And the best way for me to study is actually to read my notes out to someone, or to just read them out loud, so that's what I'm going to do. Oh! Okay, my audio back. So, starting out with just the simple people behind the developmental biology world is, oh, actually, let's do this in a darker color, is Jean Perrin. Oop. He won the 1926 Nobel Prize, and he actually proved Einstein's theory of matter correct. His famous little quote is, "Scientific advances equal," well, they explain they explain the complex visible with the simple invisible. So, like atoms and stuff like that, the simple things such as an atom explains stuff like complex life forms like us. And so there's also this biological revolution, genetics, and I'm going to abbreviate developmental biology as DevBio. All of these things kind of go together. Without one, there is no other. And by evolution, I mean microevolution, like simple, tiny changes in genetics that drive changes in organisms. And pretty much this explains how forms are made. And technically with all life, well, pretty much with all life in general, this is what I have in my notes. There are only 250 to 350 cell types. So, actually, I should have this on a different layer so I can do that. And so, we start out life, all organisms start out life as a zygote. Well, most organisms that reproduce with the fusion of the egg and the little egg and, the little sperm. The union of these two is what creates a zygote. And so, yes, there is going to be different cell talks, but I'm not displaying anything bad like that. And so, zygote goes to an adult human, which has 37.2 trillion cells, which I think that's just bonkers. In the adult human, that's how many cells we have. And then, of those cells, there are 86 billion neurons, and neurons are our nerve cells. And so, whoa, that's not what I wanted to do. And so zygotes, they organize, they repair themselves. My R's look like crap. They look like weird C's. But they repair themselves. They regulate themselves. And they can signal. And all of this right here is due, once again, is due to genetics. So genetics contributes to all of this. The zygote organizing, repairing, regulating, and signaling itself, but also to trigger it to become an adult human with that many trillion cells. So let's talk about the people behind developmental biology, or people that contributed to it. Hippocrates. He lived in the 5th century BC. He's known as the father of medicine. And he was pretty much the one to coin the term primum non nocere, which I've probably been pronouncing that wrong, but it means first do no harm. And that's pretty much what medicine does. That's what medicine is all about. You do no harm to someone, you try to help people. Though, then, along with Hippocrates, we had Aristotle. He lived in the 4th century BC. He was the father of science, which also he was the beginning, he had pretty much the beginning of embryology, which is the study of embryos. And he was one of the first people to bring up the question, do parts emerge together, or do they emerge in succession? And so that's where that started to be wondered way back before the time of Jesus. And yes, I am a Christian, so don't, sorry if I'm saying that, but, well, actually, I shouldn't apologize for that. Next, about 2,000 years after these guys, we had the term epigenesis beginning, which that is the term to say that the embryo develops progressively from an undifferentiated egg. But unfortunately, for about 2,000 years, people thought that all life was preformed, which means the embryo developed from a preformed version of an organism. So, like, we have our person. People who were involved with preformation thought that, in human females, or AFABs I really should say, people with ovaries. Inside the ovary, there was an egg with a little preformed person inside, which, of course, that's obviously not true, but that's what they used to think. William Harvey is our next person on the list. He was in 1651. He was actually the first to identify the egg as the basis of life, which began the ovis, because the ovum is actually what the egg is. Its proper term. That's what it's called. And then Robert Hooke in 1655, he coined the term cell, because he actually looked at cork through like one of the very early microscopes, and he saw that they had cell walls and they looked like little cells, and so that's why he called them cells. Because if you look at a cork cell, it very much looks like a cell. It looks like, like a jail cell or something like that. And now we have Marcello Malpighi. He lived in 1660. He was actually a Czech embryonologist, and he proposed parthogenesis. Which, do I have that written down somewhere? I somehow don't have that written down. I'm going to Google that real quick, because I'm like, how am I supposed to know what this is? Parthogenesis. Parthogenesis. That was parthenogenesis. Oh, okay. Parthenogenesis is basically it's basically reproduction from an egg without fertilization, which apparently some animals can do, so that is actually a thing that happens. Just not very often, obviously. In 1670, we have John Sommerdam. He actually popularized the model for preformation, and he claimed that the first organism contained all the future embryos. So basically, the first ever life form on Earth was probably a little bacteria cell with just a little tiny bit of DNA inside. It somehow had all the organisms that would ever exist inside of it, which that's impossible. So, well, that's gone away, because that's not true. But he was the first to actually propose that, and unfortunately some people actually believed him. But then in 1677 we have Anton van Leeuwenhoek. He was the father of microscopy. He actually was the first to describe red blood cells, and he just studied some pond water, and he discovered like protozoans, bacteria, and he actually saw spermatogenesis happen. He studied different little organisms that, would produce those, and he saw it, and he called them animalcules, or what he called little bacteria, basically. Though he, unfortunately, was also one of the people. These two groups actually existed. I'm not kidding. They actually existed back then. And these guys believed that there was actually a little tiny person just, hanging out. And yes, that is legit a drawing that somehow existed back then, but, whatever. That's not true either, so we don't like to talk about that. Well, we talk about it, but we pretty much talk about saying how it's not true. In 1840, we had Matthias Schleiden, who was a botanist, and Theodor Schwann, and who was a zoologist, they developed cell theory, which pretty much means life is made up of cells, and cells are the smallest unit of life, and cells give rise to other cells, which that is true. In 1815, there was a guy named Lamarck, and he was pretty much saying he was trying to study the inheritance of acquired characters, how people go from simple to complex organisms like giraffes and X. And then, of course, in 1859, we have Darwin, which we all know what Darwin did. On the origin of the species, descent with modification and natural selection. Which natural selection does happen, but he also proposed pangenesis, which pangenesis is how he thought like character changes occurred, which he proposed that in the environmental causes. Wow, I cannot draw a liver. There we go. He proposed that, come on, that's fine. He proposed that environmental causes caused stress on the liver, and then the liver sent out little granules, and those would cause a change in gene expression, which means you would change something in your genome that would pretty much, change what you look like, or change how you react to stuff, which, again, that's not true. So now we have, from 1866, Ernst Haskett. He popularized Darwin, but he also proposed that the nucleus contained the units of inheritance. And he also proposed ontology recapitulates phylogeny, which pretty much means the developing organism repeats evolution. And so basically, he thought that pretty much every organism goes through the lower. He thought that people, or like all organisms apparently, in utero or in development, go through the lesser, like the lesser organisms. Like if you are, wow, I cannot draw tonight. Like if you're a bird, he proposed that like a bird during development would go through the same development as like a worm, and then maybe like a fish, and then maybe some other stuff, and then it would eventually become a bird. But that, well, he was on the right track. Let's put it at that. He was on the right track. All organisms do kind of start out developing in the same way. However, that's not actually what happens. In 1893, August Weismann, he had the germplasm theory, which means germ cells are actually different from somatic cells, and germ cells are basically the cells that will eventually fuse together and become a new person, or a new organism. Wow, that face. He actually opposed Lamarck of how acquired characters happen, because he pretty much studied a whole bunch of, he studied mice. And basically, he was like, okay, that's a tail. Let's chop off the tail. And they had more babies, because, mice reproduce really fast. However, he disproved Lamarck by the mice that had had no tails because they were chopped off. Their offspring had tails. So that pretty much disproved Lamarck of acquired traits end up being passed on into offspring. Now in the 1990s, Francis Collins is the head of the National Institute of Health. He developed the Human Genome Project. And he actually found out which genes contribute to cystic fibrosis and Huntington's disease, which I think is pretty cool. And so, now we get into the actual different kingdoms of life. And so, the kingdoms of life are Animalia, fungi, Protista, and Monera. Monera is actually, well, all of these are actually eukarya. These are all part of the eukaryotes, so we don't include bacteria in these. These guys right here are monophyletic, which means they all have a very similar ancestor. Protista, the protists are actually more related to other kingdoms. Like, some of them are monophyletic with these guys, and some of them are monophyletic with these guys, which, that's why they're kind of just grouped in there. And then Animalia, that kingdom is actually a metazoan, which means that the embryo develops into 3 germ layers, which we'll be talking about maybe in a later stream, or maybe we can get to this one. Dogs, why are you growling? Dogs are growling for some reason. Okay, so there are 6 stages to life/development. There is fertilization, which is the union of gametes. And then there is cleavage, which is pretty much rapid cell division, which leads to another form of an embryo called the blastula. And cleavage, you know what cell division? Cell division leads to daughter cells. However, with cleavage, this is cell division without cell growth. So, normally a cell would grow before it divides, right? It's literally just the cell dividing. It still replicates its genome because it needs a genome to actually function properly. But cleavage is quite literally just the cells dividing over and over and over without growing. So they don't go through that G phase at all. They just keep on dividing. And then there is gastrulation, which cell division actually starts slowing down, so all of this slows down. Then the cells actually start migrating, and that's where we start to get our germ layers. So we have our 3 germ layers. And then we have organogenesis, which is where those 3 germ layers start differentiating into the different tissues and organs. And that's also the cause of morphogenesis, which is organization of tissues as well. And those are caused by morphogens, which are pretty much hormones and other types of compounds that can cause cells to differentiate. And then we also have metamorphosis. I'm going to call it metamorph. And that's the maturation of a larva to an adult. And that's also where gametogenesis begins as well. But sometimes it can also take place during organogenesis as well. But typically it's in the metamorphosis stage. And then finally we have senescence, which is the reduction in cell division as the adult actually ages. And so gametogenesis is basically, the entire purpose of gametogenesis is to create cells that actually have half the number of chromosomes. And I abbreviate chromosomes as exomes because when chromosomes come condensed down, that's quite literally what they look like. They just look like X-shaped bundles. And so that's the process of meiosis. And it occurs in the gonads, which, yes, that's what they are. But they're actually called primordial germ cells. They're actually set aside during gastrulation, and they are called totipotent, which means they can become any type of cell. They can become literally any cell type. They can become skin cells, they can become stomach cells, they could become heart cells, brain cells, bone, muscle, any type of tissue you can think of. And they actually, what these totipotent cells do, they migrate towards the posterior region of the developing embryo towards the future gonads. Okay. Yeah. So brief little educational tidbit here. Yeah, primordial germ cells, they are totipotent, so they can become any type of cell. Oddly enough, the chromosomal differentiation is somewhat different depending on what the type of animal is. So for mammals, they have XX and XY. In birds, for AFABs, they actually have ZW, and then for AMABs, they actually have ZZ. And for AFAB and AMAB, I mean AFAB meaning assigned female at birth, so assigned female at birth, and then for male, assigned male at birth. But I'm saying AFAB and AMAB for this. And then flies are even funnier. They have XX, and then they can either be just one X or they can be XY. It depends on the species. For wasps, wasps are strange. That's all I'm going to say. A fertilized 2N egg will become AFAB, and then the unfertilized 2N egg will become AMAB, which, again, it's like I said, wasps are weird with their genetics. And then you have primary determination, which is pretty much the determination of what types of gonads one is going to develop, and that's determined by like XX or XY. And no, we don't talk, in this class, we only mainly talk about the quote-unquote binary sexes, which I know there's more than just male and female sexes, obviously, but we don't really learn much about that. But with mammals, XX, XY, the reason that gonads become testes are because of the SRY gene, and then for XX to develop ovaries is the WAF4 gene. And these are both genes, oddly enough, which I think is actually pretty cool. And so, ovaries, they have two types of cells. They have granulosa cells and theca cells. And so what granulosa cells do, they produce estrogen and progesterone. And what theca cells do, they pretty much stimulate the Mullerian duct, which pretty much lead to the development of the uterus. Yeah, I know, I have to go through all of this stuff. I'm not going to remember it if I don't. It's somewhat awkward, I can probably guess. But, oh my gosh, I keep hitting my arrows. as you can guess, these organs also have particular cells. Sertoli cells actually produce AMH, which is actually anti-Mullerian hormone, which blocks this development. So it pretty much prevents this from developing. Oh, excuse me. And then the Leydig cells produce testosterone, which we all know. We all kind of know what that does. So, meiosis, yeah, the primordial germ cells, they start out as 2N, so they have 2 copies of each chromosome. In meiosis 1, they go through a division. So, we have a cell. In meiosis 1, it duplicates its genome and then divides. But at the same time, this is prophase 1. This is, in meiosis 1 is where crossing over can occur, so 2 of the same chromosome can actually kind of split and swap bits of their chromosomes, and that's how you can get more genetic diversity. But then you have meiosis 1, and then those 2 cells divide in meiosis 2, where you get half the amount of chromosomes. And so that's how those are developed. Okay, I know. People are probably going to be like, well, why don't you talk about it? Because, well, you know what? I'm going to skip over it. Because I'm going to skip over it. Because I know how weird people can be on YouTube, and I'm just going to skip over it. I'm looking at what type of stuff I can talk about. Okay, let's talk about the after fertilization occurs. So, okay. So, we have our egg, and we're talking, an egg actually has an animal pole and a vegetal pole. Vegetal pole pretty much is all of this nutrient-filled area, while the animal pole has all the genetic information. When the sperm fertilizes the egg, oddly enough, there's a 30-degree kind of rotation, almost. And that actually is where, right here, this thing called the gray crescent develops. And the gray crescent, it's pretty much showing like where the dorsal side of the organism is going to be, because you know, you have like a fish. This is the dorsal side, this is the ventral side. With a human, drawing them from the side, arm, legs. This is the dorsal side, and this is the ventral side. So it's pretty similar, just imagine the organism being upright. So the gray crescent forms, and that determines the dorsal and ventral side of the organism. Is pretty much where the sperm enters. And oddly enough, and actually, so if I draw this, where the gray crescent is, so this is this back here. The first division after fertilization is actually right here. It's front to back, and so it determines the left and the right of the embryo. Girls, stop growling at each other. Tasha, stop. Ay. But the zygote, this is a zygote where it's fertilized. But then once it actually then goes to a 2 cell, which is this, and then a 4 cell, 8, 16, 32, 32, and at this stage, the bundle of cells is actually called a morula, and then at 64, 128, that's when it's called the blastula. And at this stage, you actually start to see the development of blastomeres. And there are 2 different types of blastomeres. There's the macromeres and the micromeres. Macromeres are typically bigger cells, and they're more on the vegetal pole side because it's just mainly yolk, basically, like, you know, egg yolk. It's kind of like that. It's basically just nutrients. And then with the animal pole, we have the micromeres, which are a lot smaller, while the vegetal pole stays rather large in size with their cell divisions. And so cleavage, again, is cell division without cell growth. And so basically, it's pretty much just the cells getting smaller as they divide. Now and here's a fun fact. The frog embryo can create 37,000 cells in less than 2 days. That's a lot. And then the fly, their embryo can produce 50,000 cells in 1 day. That's how quickly this happens. And there are some cells that are determinate, and there are some that are indeterminate. Determinate cells lose their totipotency after the first division. So once this division right here happens, boom, the cells are already not totipotent, so they can't become any type of cell they want. Indeterminate, they actually lose their totipotency around the 8-cell stage. So about something kind of like this. So it's about right around here where they actually start losing their totipotency. But that's not all organisms. Determinant, these are also the protostomes. And protostome means mouth first. Proto means first, stome means mouth. These are like your worms and some other organisms like that. And then the deuterostomes, which are most vertebrates, so pretty much all organisms that have like some sort of a spinal column with the bones. These are deuterostomes, which means their mouths form second. So you can probably guess what forms first. And so the deuterostomes, now we get to start working with the colors. The deuterostomes here, oh gosh, the yellow you can't even see. So just like in class, the yellow is going to be like a golden yellow orangey kind of color. Because these are the colors the 3 germ layers actually become. And so in deuterostomes, this is the gastrocele, the beginning of pretty much all the digestive organs. And obviously, the mouth forms second. So this area will eventually become the mouth when the gastrocele comes all the way through. And this is the other end, obviously. With protostomes, because this is a deuterostome, in protostomes it's a lot different. Well, it's pretty similar, but the development is still slightly different. We still have the same kind of principle here at gastrocele. However, we have a little bit of a splitting off with the gastrocele, and that actually starts to form this red layer right here. It's the same kind of principle, but obviously forms slightly different. Now there are 3 different types, actually 2 different types of division. There's meridonial, which is along a vertical axis. It's a longitudinal cut. It actually cuts mid-sagittal, animal pole to vegetal pole, and it cuts left and right. So like I showed before with the gray crescent right here, that's a meridonial cut. It cuts like this. And it mainly just cuts down the middle. If you have an equatorial cut, it's quite literally like that. Or if you want to show the gray crescent over here, it cuts like this. And it's equatorial, so it's equal. It can actually be equal and unequal, so it could be like boom, 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 and then only boom over here. But it's pretty similar to that. And then there is actually holoblastic cleavage and meriblastic cleavage. And so holoblastic cleavage is a complete cut through the cell. So this would be the cell that completely divides. However, meriblastic is only half. it doesn't fully extend. So this would be a case of with the animal pole and vegetal pole, the division only goes through the animal pole and not through the yolk, because the yolk actually is a lot thicker, and so it's a lot harder. It's thicker and it's got a whole bunch of different proteins and nutrients, and so it's harder for cell division to go through there anyway, and plus it's not as needed. And so now I'm actually going to show how the cell divides during cleavage. And so this is the gray crescent, of course. The first division is boom, right like this. So animal pole, vegetal pole. The first division is boom, right down the middle, splits left and right. However, the second division, here's our gray crescent again. This is the first division, so left and right. The second division is boom, like this. It splits anterior from posterior, or posterior, however you pronounce it. I say posterior. The third division is an uneven cell division, and it splits the cells up here. And so now you can see the micromeres and then the macromeres forming right here. And so then the 16th, or the the fourth division to make a 16 cell is like this. Whoop. So all of these cells are dividing. So you still have macromeres and macromeres, but you can see how it's starting to split up. And because all of these cells are dividing, you have a division here, you have a division here, division here, and these three happen. Well, actually, let me boom, boom, boom. The red divisions happen all at the same time. That's how you know that it's pretty much like this. And that this is where it's called a morula, which means it's the 16th cell. And that's also when the blastocele starts forming. And the blastocele is basically a cavity. Well, the 64 cell is when the 64 cell embryo is called a blastula because that's when the blastocele starts forming. The blastocele is a cavity that forms. And so, ooh, I'm on the wrong, there we go. So imagine the thing is cut in half. So the bundle of cells is cut in half. This is what we would see on the inside. We would see this hollow area right here called the blastocele. This is the vegetal pole, so a lot of yolk would be in here, and then animal pole. This pretty much provides a cavity for cells to migrate actually inwards, and then gastrulation actually starts occurring at this point. Once it becomes like this, that's when gastrulation starts occurring. And this is when cell migration actually starts occurring, and the cells start differentiating into the germ layers. And so we have 3 germ layers: ectoderm, mesoderm, and endoderm. They are actually classified by these 3 colors. I did not pick them because I wanted to. That's actually what they look like. Oh, that's what they're pretty much labeled by. Ectoderm pretty much forms your skin, and so it also forms your central nervous system. The mesoderm forms your muscle, connective tissue, blood, all of that stuff. And your endoderm is your digestive organs. So and even also like your lungs, your liver, your pancreas, all of those inner stuff. Whoa. Okay. Yeah, and mesoderm also forms like your bones and your gonads, all of that stuff too. Your mesenchyme, which mesenchyme I'll show later. So now we get to start showing the pretty stuff. Well, kind of pretty if you consider this pretty. These will not be pretty pictures, I can tell you that right now. No, let's do yellow first. technically it's yellow, but I'm using an orange so you guys can actually see it. So all of this color coding, this is all endoderm. And all of this is mesoderm. And so, but it'll pull, and we'll pull. And so this right here is the dorsal lip. This is the ventral lip, because before we had the splitting showing the dorsal and the ventral. Well, actually, be more like this. But this is now called the Neuro Seal, which pretty much is the beginning of the digestive tract. That's what the Neuro Seal is. And this is where you start to have pretty much this, it's called epiboly, and it's where the cells actually start migrating around. So all of this ectoderm cells, they start migrating around, and then they enfold in on themselves, and this enfolding is called invagination, and it's pretty much making a hole, basically. And basically where that blastocoel was, the gastrocoel comes in and takes its place. And right here you have a whole bunch of yolk endoderm. The yolk pretty much becomes endoderm. And this is the ventral lip. I'm trying to look at all my little notes here. Yeah. But gastrulation is pretty much the most important stage in development, because without it, we would pretty much be permanent blobs of cells, and I don't think we would like that very much. Okay, I do know I need to know this. So we have a fish. This is pretty much just showing you here. So a sagittal cut is pretty much left and right. So if I were to split this fish in half, left to right, there'll be half of it on one side, half on the other. That's called a sagittal cut. A transverse cut would be something like this, just up and down, anterior to posterior. Well, it shows, and it splits it between anterior and posterior. This is anterior, posterior, dorsal, ventral. And then a horizontal cut splits dorsal and ventral. And that's just more helpful for people in this class that are learning these. Okay, what do we learn? Let's skip that. Okay, so neurulation. Neurulation it takes place still during gastrulation, but it's the morphogenic induced events that pretty much transforms ectoderm into the neural tube. And the neural tube is pretty much your very, very, very early spinal cord. And so let's draw what happens. And this is, wow, that's terrible. That's not much better, but you know, whatever. Actually, boink. No. So glad I can reverse this. That looks so much better. Okay, so here we have our developing embryo, and it is a transverse cut, so, you know, just taking down the middle of something, like that. And we have our ectoderm right here, and now we're introducing a new little organ that will unfortunately disappear eventually, but it is called the notochord, and it's actually mesoderm derived, so it's derived from the mesoderm. Forgive my sniffles. So we have our mesoderm right here. Also forgive the sketchiness of this. I'm not going to color this in because lazy. So we have our mesoderm right here. And then we have on the inside, whoa. Now I am going to use my line work layer. Wait, what size is my, oh, that makes more sense. So now we have our endoderm right here. This one I will kind of squiggle in. Show that yes, all of this is mesoderm or endoderm. But when it comes to this, the notochord, it's mesoderm derived, but it pretty much tells the ectoderm to start forming this neural lip right here, or these neural crests right here, and they actually start forming a neural plate underneath. So you have the basic layer of ectoderm right here, but then you have this plate right here, this neural plate. And this little guy right here, it pretty much starts secreting hormones to tell the ectoderm to start folding up like this. So it forms these little lips. And so eventually you have this big, like this big groove forming because of the notochord telling it, hey, fold in on yourself. And this is called the neural groove, and this is still called the neural crest. And you have these little bits right here. These are called the neural crest cells right here. And what these guys eventually do, so you still have your ectoderm right here, but now what you have is you have a tube of ectoderm cells with a little bit of the neural plate here. And this is called the Neuro Seal. And pretty much this is the beginning of your spinal cord. This is what eventually turns into your spinal cord. And sometimes there's a secondary event. Sometimes the cells will start out closed and then they'll open up. But most of the time it actually starts out just like this. Sometimes it does this, but pretty much majority of the time it forms just a tube on its own. But when you look at an organism very early on in its development, obviously, pretty much what you see with this neural tube, because most organisms have a brain, you see a thicker band of cells near the cranial region. And these little bits right here, the little holes are called neuropores. So this is the cranial neuropore, and this is the caudal neuropore. And this is all pretty much because of the notochord telling it what to do. So now we're getting into some a little bit more interesting stuff here. I'm going to use my higher stabilizer on this. So we now have our developing embryo. There's the notochord. We're starting to get these little divisions in the mesoderm. I'll tell you why here in a second. Let me get these drawn. Boop. Then of course we still have the endoderm. So now that we're here, these splittings in the mesoderm and these splittings in the mesoderm right here, that is because they're going to become different areas. So this is epimere, these two bits right here, and they are the paraxial, and they actually segment into somites. And the somites are pretty much just going to become like the vertebral column, the ribs, but then they also become like the dermatomes, which connect to skin, and then the myotomes, which actually become skeletal muscle. So that's what the epimeres do. Oops. Undo. There we go. Then we have right here these red squares are called the mesomere. They become the kidneys and the gonads and stuff like that. Then we have the hypomere, which is the lateral plate mesoderm. This entire thing right here is pretty much called the lateral plate. We have the coelom. They develop into the coelom, which is like the body cavities. And so there's like the lung cavity, the heart cavity, and just the abdomen. So this would be like the muscles surrounding your intestines and stuff like that, like your abs and stuff like that. And then the pleura, which is the lung cavity, which the lungs are actually formed by endoderm right here, but this is just like the protective layer that goes around them, like your rib cage and stuff like that, and the muscles surrounding your rib cage. And then the peritoneal is where it protects your abdomen, and the pericardial is where your heart actually like starts developing and stuff like that as well. And then you have somatopleuric cells in here, these are actually associated with the ectoderm, and like I said, mesoderm is connective tissue. And so the somatopleuric connects the mesoderm with the ectoderm. And then there's splantopleuric, or pleuric, which is associated with the endoderm, and that actually forms like the heart and the circulatory system. So the splantopleuric is the type of cells that connect here. And so, let's see, do we want to draw a very early embryo? Yeah, let's do that. Let's draw a very early on embryo. Again, I'm going to have to use this. Because like I said, the stabilizer helps so much. Okay. Lighter blue for this one. Not light enough. And then notochord here. Yellowish. Whee! Whee! Bloink. Okay. Oh, I need more red. I need more red to put some here. Okay. So now we're getting into more of an actual sort of organism starting to develop. This is the skin ectoderm right here, this dark blue outline here. This over here is the development of starting to develop a brain. So this region right here is called the rhombencephalon. This is called the mesencephalon. Then this is called the prosencephalon. So these will slowly start developing into different parts of the brain. The notochord is still doing its job, saying, hey, develop into neural tissue. This is the head mesenchyme, which will start developing into the skull and the muscles surrounding it. This right here is called the heart mesenchyme, and you can probably guess it develops into a heart. Happy Valentine's Day, everyone. For those who are watching from places where it's actually tomorrow. And this is still yolk. All of this is just yolk. And then this is called the proctoderm. And in invertebrates, this becomes not the mouth. So this right here is actually the digestive cavity. This right here is the pharynx region, which will eventually open right here and become the mouth. This is actually the liver diverticulum. So this part of the endoderm folds in on itself and starts to become the different digestive organs outside of the stomach and the intestines that help with digestion, like the pancreas and the liver. So this right here is the foregut. And so this would be like your esophagus to your small intestine. This would be your midgut right around here, which is pretty much your small intestine. And then this right here is the hindgut, which would be your colon and your rectum area. Okay, let's see. Do I want to draw? Actually, I think I will actually leave the stream here. A lot of the stuff after this is basically drawing a lot of the same tissues, just more and more developed. So I might do that. I might actually do that tomorrow night, just because I still have stuff I need to go over. But a lot of it is repeating the same stuff, so that's one of the reasons why I'm still fairly okay with it for right now. Probably tomorrow night I will actually go over some of the more cool stuff. But yeah, so that's going to be it for the stream. Thank you so much for watching and coming to learn about developmental biology with me. And I did get some drawing in, so it is obviously an art form in itself, even if we're just drawing diagrams of the different things. Hope you guys enjoyed, and I'll hopefully see you probably tomorrow. If not, I'll see you guys next lab. I'm sorry for, well, first off, I'm sorry for not posting more. Obviously, I have exams that have been pretty much coming up a lot, and so I've been working on those. I have 2 like introductions to papers due next week, and then I have 2 exams this coming Friday, so I'm like, But yeah, I'm just trying to get stuff done. I might do something in a couple weeks for just a post on YouTube. I mean, this is obviously going to go up on YouTube, so it'll, hopefully generate some views. But this, for right now, because I'm so busy with assignments and stuff, I'm not going to be posting a lot. I am on track to graduate this semester, so in May, so I'm going to be really focused on that. I'm going to try to do really well in all my classes, so hopefully I will be able to do something a little bit more with my channel. I mean, I don't plan on making this like a big career thing. I think I'm mainly doing it for fun, but I'm glad that people, at least some people, are actually kind of watching it. But yeah, I'm definitely gonna try to do some more stuff on YouTube. Maybe some speed draws or something like that, or or like maybe some drawing tutorials or something like that, or just some speed draws. I'll see if my brain comes up with something. But until then, I'm going to be working on school stuff mainly, so I will probably be a little inactive when it comes to posting, but I will try to be more active soon. So hope you guys enjoyed, and I will see you guys later.